Hello everyone, welcome to my slide presentation. Today we will discuss about the plant tissue culture media. So what are the major constituents of the plant tissue culture media? And they are salt mixtures, they are organic substances, they are natural complexes, they are inert supportive materials, and finally they are growth regulators. So what is the macronutrient salts? The ammonium nitrate, potassium nitrate, calcium chloride 2H2O, magnesium sulfate 7H2O, potassium biphosphate, and then sodium biphosphate 2H2O, and then ammonium sulfate, and these are the major micronutrient salts. And what's the necessity of this macronutrient salt? They are nitrogen influence the plant growth rate, essential in plant nucleic acid, DNA, protein, chlorophyll, amino acids, and hormones. Phosphorus abundant in meristematic as fast growing tissue, essential in photosynthesis and respiration. And then potassium, necessary for cell division. Meristematic tissue helps in the pathway for carbohydrate, protein, and chlorophyll synthesis. And then calcium, which is involved in the formation of cell walls and root and leaf development, participates in the translocation of sugars, amino acid, and ties up oxalic acid. Magnesium, involved in the photosynthetic and respiration system, active in uptake of phosphate and translocation of phosphate and starches. And the micronutrient salts, what are the micronutrient salts? Hydrogen bromide, manganese sulfate 4H2O, zinc sulfate 7H2O, potassium iodide, sodium molybdate 2H2O, copper sulfate 5H2O, cobalt chloride 6H2O, manganese sulfate H2O. And now what are the iron sources? Iron sources are the ferrous sulfate 7H2O and sodium EDTA and 2H2O. Iron involved in respiration, chlorophyll synthesis, and photosynthesis. And now micronutrient salts. Sulfur, involved in the formation of nodules and chlorophyll synthesis, a structural component of amino acid and enzyme. Manganese, involved in regulation of enzymes and growth hormones, assisting in photosynthesis and respiration. Molybdenum, involved in enzymatic reduction of the nitrates to ammonia, assists in conversion of inorganic phosphate to organic form. Zinc, involved in production of growth hormones and chlorophyll, active in respiration and carbohydrate synthesis. Boron, involved in production of growth hormones and chlorophyll, active in respiration and carbohydrate synthesis. Copper, involved in photosynthetic and respiration system, assist chlorophyll synthesis, and used as reaction catalyst. Now the organic compounds, carbon sources and our energy sources, they are the sucrose, sometimes glucose or fructose. Plants need carbon, we know that. Such as vitamins. As vitamins, there are thiamine, hydrochloric acid, pyridoxine, hydrochloric acid, nicotinic acid, glycine, and that's it. And there are still other organics such as organic acids, like citric acid 150 milligram per liter, typically used with ascorbic acid, which is used as 100 milligram per liter as an antioxidant. Phenolic compounds, fluoroglucanol, which stimulates the rooting of shoot sections. Now the natural complexes, such as coconut endosperm, tomato juice or orange juice, yeast extracts, also malt extract can be used also. And that's it. Charcoal, activated charcoal is used as a detoxifying agent, detoxifies washes from plant tissues, uh, impurities and absorption for quality vary, and concentration normally used is 0.3% or lower. Charcoal for tissue culture, acid washed and neutralized must be done and never reuse. Now the growth regulators or growth hormones. First of all, auxin. It is used for root induction and cell division, cell elongation and formation of calyx. Cytokinin is used for shoot formation. Gibberellin, it is used for cell enlargement and internodal elongation. Abscisic acid, plant stress hormone, either stimulate or inhibits callus growth depending on the species, normally used for the induction of embryogenesis. And that's it. Now auxins, callus formation, rooting of cuttings and induction of the adventive embryogenesis. IIA, indole 3 acetic acid, naturally occurring. On the other hand, IBA, indole 3 butyric acid. On the other hand, NAA, naphthalene acetic acid. And 2,4-D, 2,4-dichlorophenoxy acetic acid. And 2,4,5-T, this is 2,4,5-trichlorophenoxy acetic acid. These all are the types of acetic acid. Now cytokinins, enhances adventitious shoot formation. There are also some types such as BAP6 benzyl aminopurine, BA6 benzyl adenine, and 2-IP dimethyl aminopurine, naturally occurring. It is kynetin, furfuryl aminopurine, and GRT, naturally occurring. That's it. Gibberellin now, occasionally used in tissue culture. In some species, these hormones are required to enhance, and in others to inhibit the growth. GA3 is the most common gibberellin. Enhance callus growth and induce the dwarf or stunted plantlets to elongate. And that's it. Now abscisic acid, primarily a growth inhibitor, but enables more normal development of embryos, both zygotic and adventitious. That's it. Now as a solidifying agent, we use agar 0.5 to 1% in culture media. The pH level must be 5.0 to 6.0, generally 5.8, and the media must be sterile. Autoclave at 15 psi for 15 minutes. So commonly used plant tissue culture media are MS medium, which is known as Murashige Skoog 1962, LS medium, Linsmeyer and Skoog 1965, B5 medium, Gamborg et al. 1968, Nischt medium, Nischt and Nischt 1969, N6 medium 2 1978, E1 medium, Gamborg et al. 1983. Now the composition of MS medium required for in vitro culture. Here the left sides are the chemical constant and the right side indicates the amount of the constituent. First of all, as a macronutrients, we use the ammonium nitrate 1650 milligram per liter, potassium nitrate 1900 milligram per liter, calcium chloride 2H2O 440 milligram per liter, then magnesium sulfate 6H2O 370 milligram per liter, potassium biphosphate 170 milligram per liter. On the other hand, we can use micronutrients, which are the manganese sulfate 4H2O, zinc oxide 4H2O, or then sodium molybdate 2 copper sulfate 5 cobalt chloride 6 potassium iodide, and hydrogen bromate or boric acid, as you know. And on the other hand, the chemical constant may be iron source, which is sodium EDTA, and the FeSO4 parasulfate 7 and as vitamins we use glycine and nicotinic acid, pyridoxine as HCl, and thiamine HCl. And that's it for today. Thank you all for being with us. Please, if you like this video, subscribe my channel, click the bell button, and also like, share and comments on my video. Thank you.